Part A, chromosomes and cell division. Every organism must make new cells. Single-cell organisms, such as bacteria, yeast, paramecia, and amoebas, use cell division to reproduce asexually. In multicellular organisms, cell division, shown below, is necessary for the organism to grow to adulthood and to replace injured and worn-out cells. When you get a cut and lose some blood, additional new blood cells and new skin cells are produced from the division of cells in your body. Stopping to think 1. How is the function of cell division in a single-cell organism different from the cell division in a multicellular organism? In the early 1900s, scientists studying cells in rapidly growing parts of a plant made an interesting observation. They saw that just before cell division, the membrane around the nucleus was no longer visible, and little dark structures, which they called chromosomes, appeared as shown at left. When the cells split apart, the chromosomes were divided evenly between the two new cells. When a cell is not dividing, the chromosomes are long, fine strands, like a very thin spaghetti, packed into the nucleus of a cell. Before the cell divides, it makes copies of all its chromosomes so that its two offspring cells can get each a complete set. Then the chromosomes become coiled, which makes them visible when observed under a microscope. Finally, the cell divides as shown. So, here we go. Nucleus is intact, chromosomes become visible. Then the membrane around the nucleus is no longer visible, disintegrates. The chromosomes line up in a line in the middle. Then they get pulled apart. The chromosomes get pulled apart on opposite ends. And then two nuclei appear, and then the cell separates from each other, making two cells. Each cell in the human body contains 46 chromosomes. When a human cell divides, the two cells that result each contain 46 chromosomes. How can 46 chromosomes become two sets of 46? It's not magic. Each crisscross chromosome is two identical copies that are attached to each other. As you can see below, each doubled chromosome then splits during division to become two identical but not now separate chromosomes. So this would be a chromosome. Then it doubles itself to make the X that we may know of before. And then there's the two separate chromosomes when they split off. Stopping to think. What would happen to the number of chromosomes in each cell if copies of them were not made before cell division? Part B, chromosomes and sexual reproduction. The 46 chromosomes in a human cell can be sorted into 23 matching pairs, as shown below. Each chromosome looks identical to its partner, with one exception. Pair number 23, which is right down here, which are also called the sex chromosomes. Female humans have 2 X chromosomes, while males have only 1 X chromosome and 1 Y chromosome. So these are our pairs. 22 everyone has, and this is a big X, little y. So that would be a male. Soon after the 23 pairs of chromosomes were observed, scientists declared that the 23 pairs of chromosomes behave just like the genes in Mendel's model. What did that mean by that? What did they mean by that? Sex cells, sperm and egg, are formed by a special kind of cell division, in which each cell receives copies of exactly half of the chromosomes. In humans, the body cells contain 46 chromosomes, but the egg and sperm contain only 23. When a sperm fertilizes an egg to form the first cell of a new organism, the new cell has 46 chromosomes, as illustrated below. Half come from the mother and half come from the father. Stopping to think 3. Why must the number of chromosomes in the sperm and the egg be half the number of chromosomes in other normal cells of an organism? When sex cells are produced, exactly one member of each chromosome pair moves into each offspring cell. Thus, when an egg and a sperm cell come together in fertilization, the new cell has 23 complete pairs of chromosomes, or 46 chromosomes. Think of the 23 pairs of chromosomes as 23 pairs of socks. An egg cell has one of each kind of sock, and a sperm cell has the other matching sock for each of the egg socks. Only when they come together, when the sperm fertilizes the egg, does the resulting cell contain 23 complete pairs of socks. One member of each pair has come from the female parent and the other from the male parent. The chromosomes of each pair carry genes from the same characteristics, see below, but the two alleles of any one gene can be different, as you learned before. So this would be one chromosome, maybe from mom, and this is from dad, and maybe this is the allele location on the chromosome for tasting PTC. Maybe dad gave you a allele for not tasting PTC. So you would have two pairs. By comparing the microscope evidence to the work of Gregor Mendel, scientists realized that the chromosomes must carry those genes. One scientist understood the location of the genes and the way they were passed to offspring. They realized the importance of Mendel's work. Stopping to think for, consider two children with the same two parents. Would you expect them to have the same sets of chromosomes? Explain why or why not. Okay, last one. Part C, so many genes. Human cells contain approximately 30,000 pairs of different genes. However, the human nucleus contains only 23 pairs of chromosomes. Each gene is a small portion of a chromosome. Only by careful study can scientists determine which gene or group of genes is responsible for a specific trait, such as eye color or blood type in humans, or seed color in pea plants. Furthermore, the more the complex the trait being studied, the greater the number of different genes which contribute to it. Even diseases such as cancer are proving to result from the combined effects of many genes. The genes are part of a long molecule called DNA, which stands for deoxyribonucleic acid. Each chromosome contains a long DNA molecule. Sometimes, before a cell divides, a mistake is made in the gene when the DNA is copied to make a new set of chromosomes. These mistakes are called mutations. If the mutation occurs during the formation of a sex cell, an offspring that results from that sex cell will be affected by the mutation. Genes give instructions to the body. Even though some mutations don't make much of a difference, and some are even helpful, most mutations are harmful. A mutation is a gene is like a change in a word in the sentence. For example, consider the sentence, I hear that noise. The 4 letters in the word hear communicate a meaning. What if we change one of those letters? The results might include, I hear that noise, or I fear that noise. The first change makes the word look a little funny, but it still sounds the same. And the meaning of the sentence would be unchanged if you heard it spoken aloud. But the second change completely changes the meaning of the sentence. Like this change in a sentence, some mutations change the information provided by the genes in which they occur. Stop and think 5. How exactly does a mutation change the form of an organism? When do such mutations occur?